Greetings, family, and welcome to the We Are Indigenous documentary series. I am your host, Dr. Amaru Shia Ali, and we are going over part 3 of our documentary series, which covers global genealogy, global genetics, the discussion of human evolution, and where do we really all come from? Is the out of Africa theory right? Is the out of Eurasia theory right? Are blacks Native Americans, or are other groups Native Americans? We have been covering these subjects and we will complete our discussion on this today. The first section of this documentary covers something very controversial, and it has everything to do with how you see yourself. Mutations. Human evolution and evolutionary biologists use this word all the time. An example might be, there was a mutation that caused dark skin to turn to pale skin as a trait selection to better absorb vitamin D. That's absolutely erroneous. Or there was a trait selection to go from pale skin and hairy to dark skin without hair all over our bodies. A fallacy. In our discussion, we go over the 5 types of scientific mutation. And in doing so, finally, with a Thor sledgehammer, we prove that Darwin was wrong. The 5 types of mutations break down for us what is going on in our bodies, what went on in our ancestors' bodies, and how to explain how we arrived with the different phenotypes that we have today. Germline mutations, somatic mutations, frameshift mutations, point mutations, and chromosomal alterations and chromosome mutations. Are we a result of a chromosome 2 fusion from chimpanzees? In part 2, we talk about the hominid family. How did they get here? Our thesis on human biological polymorphisms explains this so that a 5th grader can understand it. The common ancestor between us and all of these apes, hominids, is us. What we are looking at is the pathology of Homo sapiens through time and space, and alterations caused by catastrophes and environmental circumstances that altered our biology. We have to change our perception of human evolution. Human biological polymorphisms. What does it mean? It means how our body changes, how we improve our body, how we digress. So evolution has the presumption that we start from a primitive state and we just keep advancing. What it does not take into account in a way that is sufficient is that we also devolve through disease, and we can pass those traits on to our children and weaken our lineage, thus devolving. Evolving and devolving are a part of the polymorphic potential of human beings. Are we the result of our chromosome 2 being fused together from 2 former chromosomes that we shared with the great ape family? Are we apes? All of these questions will be answered for us. In our 3rd section, we go over the antiquity of the human being with a focus on the American origins of civilization. This one is going to be catastrophic. The antiquity of the original people. And when I say original people, I'm talking about the kinky-haired, dark-skinned human in Africa, in the Americas, and other parts of the planet. We cover the global changes in geology and how that impacted us at a time where we weren't even supposed to be here. We prove that we were here. is solidified in this presentation. And in our last section, section 4, we talk about haplogroups, mitochondrial haplogroups and Y chromosome haplogroups. This is very important for going through global genealogy. Where do we come from? Is the story that we're given in the current times out of African Thesis, out of Eurasian Thesis, etc., correct? Or is there something that we can add to this discussion to tell with definitive knowledge and proof, yeah, we were the first people on the American continent, and not the Eurasians that came here in the 3 waves that they came here? Or how did we get to Africa if we were already here? Or what was our interaction like with Africans prior to Europeans? These are all questions that we need to answer. These are all questions that we can answer. We can use archaeology, we can use genetics, we can use the new finds in the ARNA Department of Anthropology and the ARNA Aboriginal Medical Association to bring a whole new education reality to our people. Are you ready?
Okay, it's time to go to school. Human evolution, genetics. The greatest problem we have is scientific confusion via classification. Classification confusion comes by way of using terms that the people don't understand, and a gentry of scientists who do understand can hide information that's vital to you understanding the basics of you, your past, your present, and your future. So we have renamed human evolution to human biological polymorphisms. Why are we doing that? Because polymorphisms deal with changes of all types in the human anatomy and physiology, specifically in the gene code. Evolving has the pretense that we are primitive and we develop over time into something more advanced. Yet we all know, even from our own lives, that our health can get bad. Not just in our own life, but we can pass that bad health on to our children, our grandchildren, through our genes, through our behaviors that epigenetically and genetically impact our genes. So the concept of a one-way rise to advancement hides the fact that human beings can devolve. Evolution and evolving comes with the possibility of devolution and devolving. And so understanding human biological polymorphisms allows us to go down both highways, so we can properly measure the global genealogy, genetics, past, present, and potential future of humanity with a balanced classification system. This will also allow us to see that scientists have turned human evolution into a belief system, a religion. Let us think. The concepts put forth in regards to natural selection that have grown to the concept of mutations that are adaptations that allow for new traits to come about have created great confusion. A good example of this is a dark-skinned people going into a cold climate and therefore selecting the trait of light skin, strictly based on the cold. As we go through this discussion and you listen to what we have to provide as far as evidence and facts, you will see how preposterous these claims are. My simple scientific claim is that there is not one evolutionary biologist discussing human evolution using the term mutation scientifically correct. As you study this material, you will find us to be 100% correct. After that, you have to contemplate, how does a global group of scientists working in human evolution and genetics not understand what a mutation is? Or is there a gentry that is disseminating education to a group of people who just follow? Either way, we will be able to provide you with the proper information and proper meaning of the terms so that your education will be enlightenment, clarity, and be functional for you in the study of human biological polymorphisms, so that you can understand how we evolve and how we devolve. And what does the subject of mutations have to do with the entire discussion? All of it goes back to you. The genetic diversity, the genetic repair, and an attempt to hide your biological reality as indigenous peoples, the power in it, how your biology actually works as opposed to other people's. These are very, very intense discussions. Intense in the sense of they remove all emotion and only deal with the science. And as the science is laid out, how people respond to it gives them clarity about their past, their genealogy, their genetics, and more importantly, what is the best way to respond to that reality as we learn it to make us a healthier people. Now let's discuss mutations. There are 5 types of mutations: germline mutations, somatic mutations, chromosomal mutations, point mutations that deal with things called SNPs, single nucleotide polymorphisms. That is adenine, cytosine, guanine, and thymine. And another thing called frameshift mutations that also deal with the nucleotides. None of these are hard to understand. We will define each one and return to our conversation on human biological polymorphisms and evolving and devolving. The first type of mutation we will discuss are germline mutations. They occur in gametes, or sperm and ovum. These mutations are especially significant because they can be transmitted to offspring, and every cell in the offspring will have the mutation. An example of these would be if a father drank alcohol excessively and he damaged his sperm cells. Those cells, which hold chromosomes, which are made of genes, will allow for those weakened genes through an epigenetic activity, alcohol, to be passed on to the child and can cause injury to the child. Once the egg is fertilized, that cell can attempt to repair itself of those injuries through a process called methylation. It could be successful or it could be unsuccessful. And that state of success or lack of success will determine whether that child is healthy or not healthy. Another example for women is a woman on birth control. The woman who is on birth control can epigenetically impact her ovum. The damage to the genes can be passed on to the daughter who is conceived. If during gestation the child is not able to repair the cells, then that trait can be passed on to that child, and that child could develop infertility or other reproductive problems. Somatic mutations occur in other cells of the body. These mutations may have little effect on the organism because they are confined to just one cell or its daughter cells. Somatic mutations cannot be passed on to offspring. An example of this would be, let's say we have someone with an alcohol problem again, and it damages liver cells and the genes in those liver cells. Because it is not the reproductive cells, that injury is confined to the liver, and therefore the child will not inherit that particular trait, but the individual will be injured by those mutations and will need to repair those or suffer the consequences of a damaged liver. As you can see, neither germline mutations or somatic mutations have anything to do with how evolutionary biologists teach human evolution and discuss mutations or adaptations. So we will continue with the other types of mutations. The third type of mutation is chromosomal mutation. Chromosomal alterations are mutations that change chromosomal structure. One example is they occur when a section of a chromosome breaks off and rejoins incorrectly or does not rejoin at all. Or there could be insertions, deletions, additions. All of these are chromosomal alterations. Here are some depictions of a deletion, a duplication, an inversion, an insertion, or a translocation. Down syndrome adds a third chromosome and is called trisomy. This causes a genetic disorder. Deletions on the 21st chromosome cause major brain abnormalities. Chromosomal deletions and alterations have nothing to do with human evolution, and there is only one claim in modern science where there's an attempt by scientists to substantiate that chromosomal alterations have something to do with evolution. Let's take a look. All of the great apes and monkeys have 48 chromosomes. Humans have 46 chromosomes. Here we see an example of a chimpanzee with chromosome 2A and 2B. The claim that evolutionary biologists in human evolution studies make is that human chromosome 2 is a result of a fusion of chromosomes 2A and 2B in great apes. The reason why this is ridiculous is because chromosomal mutations and alterations in nature never lead to an improved operation of traits in any animal. The only time in our history where chromosomal alterations have led to improvement in any species is through genetic modification, and that came from a more enlightened being having the scientific ability to alter the chromosomes to improve that particular species. So unless human evolutionary biologists are saying the Anunnaki actually form humans, then we are left to say that their claim has no merit, and we will detail it here. First off, at the two alleged fusion sites, there is absolutely no satellite DNA. In all chromosomal alterations, you will find satellite DNA at the fusion sites. This satellite DNA makes all coding around the fusion points inactive. The ends of chromosomes are called telomeres. These ends would have fused together at the fusion site, and we would be able to tell based on the amount of base pairs of nucleotides that these were two fusion points of telomeres, which are the ends of chromosomes. The ends of chromosomes where we have telomeres usually have between about 5,000 to 15,000 bases of DNA. At this particular site, there are only 798 DNA letters long of nucleotides. We can see here the 798 DNA nucleotides that are at this particular site. This completely destroys the idea that there was a fusion of two telomeres. Scientists know this. This is where we begin to see that there is a gentry of scientists who want to hide something from us. As we continue to look at chromosome 2 and the alleged fusion site, we find that the site is actually a place of an active gene promoter. DDX11L2 is a gene that overlaps both the intron and the exon part of the genome. Exons are a part of the genome that are coded to make proteins. DDX11L2 operates in cell signaling in the extracellular matrix, sort of like a paracrine signaling. Paracrine signaling is how cells talk to each other. It also operates to make blood cell production. It's an active gene. There is no way that we could have an active gene at a chromosomal alteration site, a chromosomal mutation site. Human chromosome 2 is not fusion at all. It's an operating normal chromosome. It meets none of the biological standards as a fusion site. So in summary, evolutionists struggle to explain why humans have 46 chromosomes and apes have 48, if both descended from a common ancestor. The supposed answer is the fusion of two chromosomes in the past. The alleged fusion site isn't connected to satellite DNA sequences like documented fusions are, and it's too small and muddled to be the fusion of two chromosomes. Most importantly, the fusion site is located inside a gene and contains intricate coding functionality, soundly refuting fusion. The alleged cryptic centromere site is inside a large protein coding gene, further refuting the fusion idea. The human chromosome 2 fusion theory doesn't hold up to scientific scrutiny. So the grade 8 family, just like those who have Down syndrome, are suffering from additions of chromosomes into their biology. This has caused deformity amongst them, and their common ancestor is actually Homo sapiens. Therefore, they are the perfect representation of devolving based on biological polymorphisms that have led to their disease states. If we peruse the planet now and we look at most of the great apes, it is humans that are keeping them alive. Their populations have dwindled significantly, showing that they do not have fitness to survive. Chromosomal alterations do not bring about improvement in any species. They are the third of 5 types of mutation that cause devolving amongst biological organisms. Our 4th type of mutation deals with point mutations and is connected to SNPs, single nucleotide polymorphisms. These are adenine, cytosine, guanine, and thymine, and point mutations in this sense deals with changes in the order or the coding of the 4 types of nucleotides. Our first type of point mutation is a silent mutation, and this occurs when a mutated codon codes for the same amino acid, therefore causing no issue. Here we have an example of a silent mutation in glutamine. The C stands for cytosine, the A is adenine, and the G is guanine. Since adenine and guanine are both purines, substituting a G causes no issue, and we get the same amino acid produced, glutamine. The next type of point mutation is called missense. This is a mutated codon that codes for a different amino acid, and herein we will have a problem, as we will see as we continue. Here we have glutamine, CAA, and we insert a pyrimidine, cytosine, CCA, and now we have a completely different amino acid. The cytosine as a pyrimidine does not match the purine groups. Therefore, we have a new amino acid with this missense mutation. Our last type of point mutation is a nonsense point mutation, and it is a mutated codon that is a premature stop codon. Here we have glutamine again, CAA, and we have a uracil inserted as UAA, which is a stop codon. which instructs the stopping of the production of the amino acid chain. This also can present a serious problem in the transcription and translation process of proteins. The important point here, though, is none of these point mutations with SNPs, silent, missense, or nonsense, have anything to do with human evolution, natural selection, or any of the theses or theories that have been presented by evolutionary biologists. Again, what do they mean when they say mutation? We are at our fourth scientific definition for mutation, with one to go, and none of them match anything to do with the definitions that evolutionary biologists are using in human evolution. Our fifth and last type of mutation is called a frameshift mutation. It is a deletion or insertion of one or more nucleotides that changes the reading frame of the base sequence. Here are some examples of frameshift. Deletions remove nucleotides and insertions add nucleotides. Consider the following sequence of bases in RNA: AUG, AAU, ACG, GCU equals start asparagine, threonine, alanine. Now assume an insertion occurs in the sequence. Let's say an A nucleotide is inserted after the start codon AUG. It equals start lysine, tyrosine, glycine. Even though the rest of the sequence is unchanged, this insertion changes the reading frame and thus all of the codons that follow. As this example shows, a frameshift mutation can dramatically change how the codons in the messenger RNA are read. This can have a drastic effect on the protein product. As we have seen, human evolution presents no concept of mutation that actually goes with the 5 scientific types of mutation. In their paradigm, fitness is the ability of an organism to adapt. That fitness allows the organism to continue to procreate and avoid disease so that the organism or species can survive. As we will see, there are explanations for why a human loses their melanin, why a human gains or loses a hairy body, fall very short from giving thorough explanations in regards to human biological polymorphisms. In fact, they only explain genetic disease. And as we continue, we will show genetic repair, genetic diversity, and genetic disease, and it all being based on the organism evolving or devolving, based on human biological polymorphisms. Now we will use the S&P networks to explain the scientific fact that there is only genetic diversity, genetic disease, and genetic repair, and that natural selection is a complete myth. In this part, we will go back to our point mutations and focus on missense and nonsense, and focus on frameshift mutations, because these types of mutations will help us understand the applicable nature to genetic diversity, genetic disease, and genetic repair. When it comes to the nucleotides, adenine is always coupled with thymine, guanine is always coupled with cytosine when we have functionality in the nucleotide order. It is the transversions we want to watch out for, when A, which is normally coupled with T, is now coupled with C, and G, which is normally coupled with C, is now coupled with T. This crossing of SNPs in point mutations were called missense mutations, and they cause the coding for the wrong amino acids. This distortion of the synthesis of proteins can cause major cellular damage and major malfunctions in the physiological processes within a cell. Or they can cause protein synthesis to stop, nonsense point mutations, and this all leads to genetic-based disease. Here we have a great example of the point in this article from Genome Medicine. It deals with UV radiation and how it causes cancer and mutations in the skin of white skin or European or Eurasian peoples, and how the DNA damage is repaired or stays in a diseased state. And it reads, cancer mutation signatures from external sources, DNA damage. A major external source of DNA damage is ultraviolet light, which can cross-link adjacent pyrimidine bases, CC, CT, TC, and TT. If such a pyrimidine dimer is not repaired and becomes a substrate for DNA replication or local synthesis, then most DNA polymerases will follow the A rule and insert two adenines opposite the dimer. Late repair or another round of replication can then immortalize the original lesion as a C to T transition mutation. Thus, the mutational signature of UV light is predominantly C to T transitions in dipyrimidine contexts. Other features of UV-induced mutagenesis include the occurrence of adjacent mutations, mostly CC to TT, in a non-transcribed strand basis due to preferential repair of the transcribed DNA strand. The science behind the nucleotides is that adenine and guanine are found predominantly in those of African descent or indigenous descent. The adenine and guanine are alkaline. The cytosine and thymine are acidic. The cytosine and thymine are heavy nitrogenous nucleotides. The adenine and guanine are heavy carbon nucleotides. As the sun is striking these nitrogenous and ammonia nucleotides, we get lesions, and therefore when we look at Caucasians or Europeans, we see that they have higher rates of cytosine and thymine in their nucleotides, and therefore we get the missense, nonsense, and frameshift mutations from the CC, CT, TC, and TT base pairs, and it explains why they cannot live in hot places with high UV light. This has nothing to do with natural selection. Notice in the article, when there was a need for repair, there was an insertion of two adenines, an alkaline heavy carbon nucleotide. This begins to show that we are dealing with genetic diversity and genetic repair abilities versus genetic disease. Pale skin is not an adaptation. It is a genetic disease. Nature proves it. The nucleotides prove it. Genetic mutations prove it. It is not a factor of humans evolving. It is a factor of genetic disease and humans devolving. This is why they hide what mutations really are. They're hiding their own reality, their own history, and giving us a pseudo theory in order to hide true science. What you are hearing now is true science. Here we have an article from the International Journal of Molecular Sciences. The MC1R gene is the gene that produces eumelanin. This article, the MC1R front and center in the bright side of dark eumelanin and DNA repair. The article discusses how dark eumelanin engages DNA repair. When the MC1R gene has mutations, it produces pheopigments, the yellow to red colors found in Europeans and some Asians. The types of mutations we described before, specifically point mutations and frameshift mutations, create a situation where the improper amino acids are produced. Here we have the signaling pathway for eumelanin versus pheomelanin. The eumelanin signaling pathway has alpha melanin stimulating hormone, which the Europeans do not make. The adrenocorticotropic hormones that are made control androgen synthesis and regulation of cortisol. The androgen synthesis increases fertility and makes for more anabolic buildup in somatic cells. Anabolic here meaning more complex molecules to make the cells more complex. What I'm simply saying to you is that a nappy-headed, dark-skinned human is more complex, more genetically diverse, and genetically superior as the original human of the planet Earth. This is what human evolutionists are trying to hide. Let us read directly from the study. Summary of the effects of the MC1R agonist, or those that promote MC1R, alpha melanin stimulating hormone and ACTH, and the antagonists, ACEP and HBD3. Antagonists meaning that they stop the production of functional MC1R, and the impact of loss of function of MC1R on these effects. Activation of the MC1R expressed on the cell membrane of melanocytes by either agonist, alpha melanin stimulating hormone or ACTH, activates the CAMP pathway, leading to increased eumelanin synthesis, which quenches reactive oxygen species. That's a free radical that damages tissue. So melanin is stopping the damage of tissue and reduces the generation of DNA damage upon UV exposure. Activation of the CAMP pathway also enhances the DNA repair and antioxidant capacities of melanocytes. So here you see melanin is considered an antioxidant, like the ones that you eat to try to keep yourself healthy. The cumulative outcome of these effects is maintenance of genomic stability of melanocytes. Treatment of melanocytes with either ACEP or HBD3 antagonizes the effects of melanin-stimulating hormone and are therefore expected to reduce genomic stability. Reduce genomic stability through pheomelanin, that is. Expression of the loss of function of MC1R disrupts the signaling of the agonist-bound receptor, thereby inhibiting the synthesis of eumelanin, allowing only the synthesis of pheomelanin, which increases the generation of reactive oxygen species and nitric oxygen species. They are telling you that pheomelanin is causing more free radicals and damage. That is not evolution. What's happening here is genetic disease. This is not an adaptation. This is genetic disease. This is devolving. You don't go to cold places and get pale skin. That's not how pale skin came about. We will talk about that. And allows for increased UV-induced DNA damage. Additionally, lack of signaling of the alpha melanin stimulating hormone-bound MC1R via the CAMP pathway inhibits the activation of antioxidant and DNA repair pathways, leading to reduced genomic stability. Y'all get what's being said here. This is clarity. This is science operating. This is another example of how red hair gets made. The arrows are pointing to valine and arginine dysfunction in the synthesis of the amino acids. So here you have again point mutations that are occurring that cause the melanin not to be placed in the hair shaft. And it's all based on point mutations. Red hair, pale skin is not natural selection. It is genetic disease. So what is at the basis of this genetic power or genetic diversity? It is carbon. Carbon is the most covalent chemical on the periodic table. It has the ability to bind with more molecules at a stronger capacity than any other element, and it is the basis of melanin. Therefore, we have the ability to have more anabolic buildup or more complex molecular structures. So this reality is at the basis of our molecules and our nucleotides. and the ability to make things like alpha melanin stimulating hormone. Here we see why carbon is the most important element. Its complexity. It has the potential for the most biological complexity. Abundance. It is the most abundant. Stability. Its shells that create covalent bonds are closer to the nucleus than the other elements that have the ability to bond with 4 other atoms. Example, silicon. Silicon has a bonding strength of 134 moles. Carbon's binding strength is 334 moles. Carbon is more versatile in building structure than any other element. The more you have of it, the more potential it becomes more complex. Carbon can make single, double, triple, or quadruple bonds. There are only 5 other elements that can do that, and none of them are as stable as carbon. Carbon is the 3rd most abundant in the solar system. It is the 2nd most abundant in the body. Carbon can make closed rings. Carbon can make long, non-repetitive polymers. It's diverse. It is a part of proteins, carbohydrates, and fats. It has the ability to carry a lot of information. It is the backbone of DNA due to its ability to carry a lot of information in long chains of molecules. It is not only the backbone of DNA, it's a part of the actual nucleotides. As shown above, it can protect the nucleotides from mutations. The driving force of chemistry is for atoms, molecules, etc. to get to an energy of efficient state. They do this via their design, which gives them purpose. Atoms want to bond, molecules want to bond, and carbon is the basis of that bonding. It builds intelligence molecularly. It is the molecular basis of our genetic diversity. We now enter our discussion on the Cataclysm thesis of biological polymorphisms. We build off of our last subject about mutations, evolving, devolving, and changes in the human lineage. Our common ancestor is the Homo sapien in this model, which at intervals of time have experienced adverse mutations that have caused great apes, other said hominids, Neanderthals, and Denisovans to come about. All of these being aspects of Homo sapiens suffering from pathological disease, devolving. None of it representing an evolution from a primal entity to a modern human being. In future discussions, we will introduce the term heterosapien to distinguish between the deeply eumelanated oldest haplogroups from the adversely mutated haplogroups that are in need of repair. This repair is a present and current study of the Aboriginal Medical Association, and we see the gene code as a self-perpetual error-correcting biological code, and that this error-correcting code is seen in nature as well, and there is symmetry between the corrections that take place on a quantum level and a biological level. We will prove that there are not multiple species of hominids, that there has been one, with diverging polymorphisms that lead to evolving or devolving, devolving through genetic disease and epigenetic disease, evolving through us learning how to maintain and repair and improve upon our genetic code. This is a current mapping of how evolutionary biologists express the development of human evolution. They have branches for the great apes, and they descend through different hominid species to get to Homo sapiens. They justify our development from these hominids based on fossils that they have found and what they see as positive selection of traits from a primitive manner expressed in the skulls and biology of hominids to an improvement through modern Homo sapiens. We have already laid the groundwork to prove that there is no positive selection and that this is an erroneous assumption that does not give a clear and concise explanation for what we are looking at in regards to the scientific facts. Here we have a map of where they have found fossils of hominids. This map will become important in the future as it will prove the Cataclysm thesis. The points at which you have hominids placed with fossils match exactly with areas of cataclysms in the past that would have caused polymorphic changes in the sense of devolving amongst Homo sapiens who were impacted environmentally by those cataclysms. Now let us look at the process of how fossils occur. Stage 1, death. In this stage, a marine reptile from the Jurassic period called a plesiosaur dies and its body falls to the seafloor. Stage 2 is key. A landslide occurs nearby and the plesiosaur is buried quickly in mud, preventing scavenging animals from gobbling it up. You will always find where there's fossilization some rapid environmental cause that buries the animal quickly. And in all of our circumstances, we're highlighting minor and major cataclysms that cause this activity. Stage 3, decay of soft parts. The muscles, skin, and other soft parts of the plesiosaur are digested by bacteria in the sediments. Only the hardest parts, such as the bones and the teeth, are left. Stage 4 deals with lithification, or the turning of the bones into rock. As time passes, more and more sediment builds up. The weight of the overlying layers squashes the soft mud and it begins to turn into rock. This is called lithification. Water is squeezed out of the mud and seeps into the plesiosaur's bones. Minerals and chemicals in this water gradually change the bones and teeth into stone. The plesiosaur is now a fossil, but it is buried under layers and layers of rock under the ocean. This process is important because now these are no longer bones. These are actually stone, mineralized, and in the shape of what the bones used to be. This is why none of the hominids' DNA can be taken and used and tested to tell whether haplogroup they are in or to match them to any of the modern DNA in Homo sapiens, Neanderthals, and Denisovans. When geneticists found the oldest DNA in the world coming out of Florida and South Carolina from black people in America, they tried to make up a story that the DNA had to be introgressed DNA from some ancient hominid. Yet, they have never taken any, I repeat, any DNA from ancient hominids. In the same way that they dismiss theists and others for their belief, it is our duty to dismiss them for their belief. Let's take a look again at where these fossils occur. Africa, Asia, Europe, and Australia. Here we have the skulls of the various great apes and alleged hominids that we are connected to according to evolutionary biologists. They use the skulls as the basis to say that the human Homo sapiens skull is different than the predecessors, and therefore this justifies our evolution. This makes no sense, as I can show here Homo sapiens with beetle brow. Yet, how did he get it? This is a case of microcephaly, a neuronal development disorder that affects the shape of the skull and the functions of the human brain. In fact, there are 7 genes that contribute to this: the MCPH1, the ASPM, the CDK5RAP2, the CENPJ, the STIL, the WDR62, and the CEP152 gene, which contribute to the phenotype that you are looking at. If his skull was fossilized and it was found somewhere 1,000 years from now, would he too be a missing hominid link? No, he would be a Homo sapien with a neuronal development disorder. Devolving, not evolving. So these fossils only represent the humans, Homo sapiens, who didn't make it. Their ability to stay fit, to reproduce continuously, was cut off. Disease, lack of resources, all contributed to their demise. We have found them at later dates, and now we are asserting that they are something that they are not. As we continue our discussion, we will go through and discuss these hominids and the locations that they were found in order to compare them to the locations geologically that underwent cataclysms. Where they were found matches exactly where these major cataclysms had impact on the human biological polymorphisms of Homo sapiens, causing them to lose their form, leaving the evidence for us to see, to witness. The fall of some of humanity, but we remained, and the evidence is in our genes, in the records left by our ancestors, in the archaeological record, in the geological record. Let's take a look. The antiquity of humanity is hidden in plain sight. America is the most ancient. This part of our series goes into the evidence to further prove what has been hidden from us. America is very ancient. And now that we have went over mutations, we can now go across the globe and measure properly through genetics, through anthropology, through archaeology, the question, where do we come from? I'll start with the basic map. This is called Gondwana, one of the phases of the early shifting of the continents. We can see South America, Africa, and other parts of the globe in this map. We do not take Pangea, Gondwana, or these early phases as the beginning. In fact, we see them as a reaction to a great cataclysmic event. Plate tectonics were not found until around the 1960s. I'm highlighting here a tectonic plate in Africa that divides North and West Africa from Central, Northeast, and Southern Africa. This division will become very, very important later as we verify the antiquity of information that has been left in the Americas. Another minor and important point is where we find the largest mountain ranges on the planet. From North America and South America, we have the Rocky Mountain Range, and all down the western side of South America, we have the Andes. We also have the Himalayas. These meeting points tell us a lot about the evidence of plate tectonics. Notice on the eastern coast of South America and all throughout Central and South Africa, we don't see these mountain ranges. Yet we see these lands connected, which actually geologically makes no sense. There should be mountain ranges where there were breaks or where there were collisions between continents. This is a map from the Ica stones of Peru, discovered by Javier Cabrera. There has been many talks and discussions about whether or not the Ica stones are authentic. Many have stated that a farmer, Ishua, made these fake stones. We have discussed this on countless occasions to verify that Yeshua stated that Javier Cabrera had 5,000 authentic stones, and that he only made reports that they were false because there was a law in Peru that stated if you sold these items, you could be jailed. Also, Santiago Calvo found stones. These stones were also found by Spanish conquistadors amongst the Inca. They are authentic. What we are seeing here is North America, South America, West Africa, and an eighth continent in the Pacific. This is depicted on stones that are thousands of years old, found in the desert, in America. Are they real? Yes, they are real. How do we know? They depict plate tectonics perfectly. Some ignorant farmers who had no idea about plate tectonics could not depict these. The eighth continent is there in the Pacific, and it was discovered that there was an eighth continent in the Pacific. We will also use NASA and JPL imaging to show that the Pacific Ocean had to have some type of ancient cataclysm. This cataclysm is evidenced by vector movements of the plates into what is called the Pacific Trench, a giant hole in the earth that we now have filled with water and we call the Pacific Ocean. Zelanda on this map is depicted here to the left with the letter B on it. Zelanda was much larger than what they are discussing now. They're only beginning to find pieces of it. And as time goes on, they're going to continue to catch up with information we already know from our American indigenous record. Here is a current depiction of pieces and parts of the eighth continent in the Pacific, Zelanda. We also see the Antan-Java Plateau. We also see the Hawaiian Islands in the middle of the Pacific. All of these areas represent the remnant areas of the loss of this eighth continent in the Pacific Ocean. Here we see geologists stating that a region of the Southwest Pacific Ocean is made up of continental crust. This study was done in 2017 discussing the same area of the crust formation found in the Pacific Ocean. Zelanda is a real continent. But how did indigenous people know this, make maps before modern science? The whole idea of evolution has us thinking that all ancient people were primitive. Here is a study from the Department of Physics and Earth Sciences at the Faculty of Earth Science, University of Ryukyus in Okinawa, Japan. It discusses what is underwater in these specific areas. A pyramid. These scientists have found a complete underwater civilization in the Pacific. Here are some of the original photos. Who built these pyramids? Where is the one place that we have evidence of anybody talking about a pyramid building civilization in this area in the Pacific? We have one source, and it is an American source in Ica, Peru. Years ago, when I used to talk about Mu from Augustus Le Plongeon, many people would laugh. Who's laughing now? In part 4 and 5 of the study, they state, there our data showed specific features looking like an ancient city beneath the sea. There are artificial features looking like a giant castle or temple, many other similar or smaller temples, a stadium, rotary, and well-organized road system connecting these structures. The stadium stands 300 miles southeast from the main pyramid. Is this a game? Or is this made up? No, it's not. These are scientists who have discovered an underwater civilization in the Pacific at the very location that Americans depicted on ancient maps. The study goes on to state Submarine research reveals the probable sunken ancient city characterized by stepped pyramidal structures and a road system connecting them off Yonaguni in the Okinawa, Japan area. The central pyramid stands under approximately 25 meters of ocean, tentatively named Yonaguni. This area has other structures. Who were these people? Where are they now? Who built these ancient pyramid systems in sunken cities that are now in the Pacific? Let us return to the Earth cataclysm that we are citing that potentially caused this. Here we see plate motion based on global positioning systems satellite data from NASA and JPL. The vectors show direction and magnitude of motion of the plates. All of the arrows are going towards the Pacific. In fact, Africa, Asia, North America, South America, and Antarctica all have vectors going back to the Pacific Trench, which is a giant hole in the earth. This says that some ancient cataclysm was caused here. Now we have the evidence, an entire destroyed continent, causing the plates to move back to their original placement. This simply also means that Pangaea, Gondwana were all phases post some ancient cataclysm on this earth. More importantly, this confirms that man was present, woman was present on this earth during this time. The millions and billions of years they attribute to Homo sapiens not being present. This presentation proves that they were. Not only did they witness these events, they left maps. Maps of what existed in the area prior to the event. They even depicted in the particular stones what caused it. Some type of object coming from the heavens. Here we see the South Americans with telescopes, and they're aimed at the heavens. And we see a streaking object coming towards where they are. Did they depict an earth cataclysm that caused what we know of as the Pacific Ocean as it stands today? How did they know that there was an eighth continent? With our modern equipment, we just found out. In fact, the discovery of plate tectonics is only about 65 years old. These are drawings of what they depicted on the stones. F is North, Central, and East Africa, which were separated from West Africa, or the mythical Atlantis, also called the first Ethiopia. E is Asia, with Europe attached, which is not a true continent, meaning Europe. G is Australia and E is Antarctica, when it was uncovered by polar ice, as it was not the South Pole at this time. Here are illustrations that depict the phases, the bottom being the original illustration from the maps from the ecostones. In progression going forward, upward, we see what we know of as plate tectonics taking place. When we go to the third progression, we begin to see the map as it is today. Once again, we see in the area where we have E in Asia, how the Himalaya mountains were formed. We also see in D, West Africa being detached. It is interesting that the Egyptians said that they were from the West, from an area of a higher civilization. Solon the priest, after talking to the ancient Egyptians, stated that they had taught him about a civilization 12,000 years ago that they came from. As we can see from earlier, F and D, which represent West and Northwest Africa, D, and F, Central, South, and Northeast Africa, were detached. This matches the current plate tectonics as shown in Gondwana, as we showed earlier. North and South America are detached, which will become important when we talk about the erroneous ocean dispersals of animals that evolution has provided to us, stating that dinosaurs and monkeys cross oceans by themselves. Did they swim? Did they take rafts? This is the preposterous information that is given to us. The missing link is man and woman, original man and woman, who were here to record these events. Not only did they record these events, the information of these events are living inside of us, in our genes, in the architecture, in the pyramid systems that they left on this planet. Now let us debunk these erroneous concepts in evolution. Ocean dispersal is a type of biological dispersal that occurs when terrestrial organisms transfer from one landmass to another by way of a sea crossing. What these evolutionists propose is that dinosaurs, monkeys, and other mammals, birds, etc., cross oceans and seas somehow as an explanation for why they show up on continents they were not supposed to be on. This violates the concept developed by the co-founder of evolutionary theory. Most of us know of Charles Darwin. Many do not know Alfred Wallace. He, Alfred Wallace, independently conceived the theory of evolution through natural selection, and his 1858 paper on the subject was published that year alongside extracts from Charles Darwin's earlier writings on the topic. It spurred Darwin to set aside the big species book he was drafting and quickly write an abstract of it, which was published in 1859 as On the Origin of Species. Wallace did extensive field work, starting in the Amazon River Basin. He then did field work in the Malay Archipelago, where he identified the faunal divide now termed the Wallace Line, which separates the Indonesian archipelago into two distinct parts, a western portion in which the animals are largely of Asian origin, and an eastern portion where the fauna reflect Australia. He was considered the 19th century's leading expert on the geographical distribution of animal species, and is sometimes called the father of biogeography, or more specifically, of zoogeography. The emphasis I am making here is the proposal of the Wallace Line. The Wallace Line was a line that separated animals of different types. They were only miles away from each other on various Pacific islands. Wallace proposed at some earlier point that the islands were separated and the animals developed independently and never had integration due to geologic divisions. It would not be until the later discovery of tectonic plates that Wallace's theory was confirmed. Animals separated by oceans and seas that developed as plates spread had no way of being on other continents. Once scientists started finding that some animals were in fact on other continents they were not supposed to be on, they made up the ocean dispersal theory. The reason this is important is because if we add in the human factor, we can simply make the logical conclusion that they were taken there by humans. Instead, these scientists theorized that animals swam across whole oceans, seas, and water barriers, even if it is known that they were land animals. This theory is not well-founded and should be done away with. Here is one of the ridiculous claims. This is where scientists abuse their positions to support flaws in a theory, evolution, that has gaps too wide to explain. Here we have a paper published in Cretaceous Research, April 2021. The first duck-billed dinosaur, Hadrosauridae lambeosaurinae, from Africa, and the role of oceanic dispersal in dinosaur biogeography. Here's a quote from the study. Here we report the first hadrosaurid from Africa from the late Maastrichtian of Morocco. We show that hadrosaurids disperse from Laurasia into Africa, despite extensive, persistent marine barriers, meaning oceans. And Laurasia in this sense was Europe. And proposed dispersal across marine barriers played an important role in latest Cretaceous biogeography. The hadrosaurid from the Morocco area was a land animal reptile and was not known as an aquatic animal who could swim long distances. How would it swim across an ocean from what is now Europe to what is West Africa, when at the time the lands were disconnected by a huge sea with no land connecting? The definitive answer is man. Let us look at another case of this erroneous ocean dispersal myth. This is from the journal Nature. Monkey teeth tale of ancient migration. Fossils found in the Panama Canal are the oldest evidence of monkeys in North America. New World monkeys such as this white-headed capuchin reached South America more than 30 million years ago. A quote from the study. A team led by Jonathan Block, a paleontologist at the Florida Museum of Natural History in Gainesville, found 7 monkey teeth in the Panama Canal Basin. The scientists have identified the fossils as belonging to a previously unknown species, Panamamcebus transitus, that lived nearly 21 million years ago during the early Miocene epoch. We never would have predicted they would have been here, says Block. The teeth are the oldest evidence for the dispersal of a mammal from South America to North America, he says. No other mammals are known to have made it across the body of water that separated the two continents in the Miocene. Current theory suggests that primates crossed the Atlantic Ocean from Africa to South America roughly 37 to 34 million years ago by rafting on vegetation. Oh my God. Could you imagine trying to raft on vegetation from South America or to South America from Africa? This is ridiculous. These New World monkeys, a group that includes marmosets and spider and howler monkeys, spread and diversified into tropical regions of South America. Scientists have thought that monkeys did not reach Central America until after the formation of the Isthmus of Panama 3.5 million years ago. The strip of land links North and South America. But Block's finding suggests that the primates crossed a water barrier and made it into North America nearly 18 million years earlier. These people are telling us two things. Monkeys traveled on vegetation from Africa to the Americas over a whole ocean. They are also claiming that monkeys traveled from South America to North Central America at a time when the two were separated by a sea. This violated the Wallace Line rule, later verified by plate tectonics. The records for man's existence in these areas is extensive. We will continue to use the best scientific facts to bring this conversation into a better place than the current theory of evolution. As we continue to look at the Earth and discover things about its antiquity, we also find that the top 11 oldest trees on Earth, 10 of them are in the United States of America. The other one is in America as well, but in South America in Chile. The oldest trees, shrubbery, cyanobacteria are all found in the Americas. This is what animals and human populations would have need to develop in order to survive. And the oldest are found in the Americas. Can we now see how the theory of evolution, the out of Africa theories, the out of Asia theories are erroneous? Let us now go to Africa to deal with this matrix of hominids, fossils, and compare it to what we have already discussed. Here we are looking at the oldest Homo sapiens in Africa at Jebel Uhud. But these were not in East Africa, in Central Africa. These were in West Africa. Here here's a composite reconstruction of the earliest known Homo sapiens found in Africa. Question: why would we not find these finds in Central or East Africa? There is a great reason why we don't. Let us go back briefly to this map of Gondwana and remind ourselves that according to plate tectonics, West Africa, North and West, are separated from Northeast, Central, and South Africa. Now let us return to our oldest world maps from the Americas, illustrations from the Cabrera Museum. We can take the separation of the continents and make sense of this whole discussion. Notice how in parts D and F, North and West Africa are separated from South, Central, and Northeast Africa by a sea. We see this not only in the fossil record where Homo sapien fossils are found in West Africa and earlier Homo sapien fossils that are suffering from microcephaly or depicted as earlier hominids are in section F. What brought about plate D and plate F, or landmass F and landmass D, coming together? It was this cataclysm that we depicted earlier, again with all of the vectors of the plates moving toward the Pacific Trench. Something happened in the Pacific Ocean that caused the eighth continent to be lost. What did the world look like prior to this period? What did the world look like after this period? We have the answers to both questions. What we know of as Gondwana was a post-cataclysmic reality. The continents slamming together. We see these plates connecting. Post this period, they drifted, as the vectors showed, back out from the site of where the cataclysm took place in the Pacific Ocean, where we had a sunken continent. If you were to imagine what's on the other side of this planet, it's a huge ocean. And now our planet, our plates, are all moving gradually towards that Pacific Trench. So now we can come back to our West African Homo sapien finds, the oldest in Africa. There's a reason that they are in the West and not the East. And as we continue this conversation, the scientific facts will come together in a perfect puzzle. If we consult the oldest writing systems in Africa, the pyramid texts, they tell us the whole story. Some scholars say that the origins of the Egyptian kings and civilizations comes from Kush in the south. I question that. The Egyptians venerated the West and despised the south, according to their own records. The origins of the falcon, jackal, and most all of the animals originate in the Americas. Pyramid text, utterance 304. Hail to you, Bull of Re, who has 4 horns. Bend down this western horn of yours for me, that I, king, may pass. Are you a pure westerner? I have come forth from the Falcon City. Utterance 81. Isis has reassembled you. The heart of Horus is glad about you in this your name, foremost of the Westerners. Bubulus antiquus, found in Tassili in Jar, rock carvings. Again, this is Northwest Africa. The animal is now extinct, but was a type of cattle in the Sahara, when it was a fertile grassland, which leads to the conclusion that the founders of Egypt, just like their records say, came from the West. But what happened to this fertile Sahara? Why is it now the world's biggest desert? This desert came about as a result of the cataclysm that we are discussing. And we have evidence that under that great desert was an entire ocean. The remains, the fossils, and the evidence are overwhelming. This is a text found by Mallet at Abydos. Some interesting things take place in this text. The Westerners, the rulers to the west of Egypt, are called Amashi. That term today has become Amazigh, but the original is Amashi, and the Egyptians were attempting to translate a word that was not a part of their native vernacular. Not only that, they used very specific and interesting terms to denote themselves. A pure Westerner was the mantra of the ancient Egyptian text. The original, Mary, meant heir. Heir to what? When it left the Canaanite and Phoenician, it went into the Greek as Meraps, the name used for the Ethiopian kings, taking on the connotation of the physical characteristics of these black kings. Here, from a text at Abydos, found by Mallet, we have the phrase Mary incept F, meaning heir of his king's nome. Mary, or Tamari, was the name of Egypt, a plow in the reeds. But we also know that this term, Mer, became the term for Moors. Now, as we talk about this discussion, we are only tracking the genealogy of peoples. I do not want to go off into what is called unscientific conversation, but at a temple at Edfu, Jut, or Thoth, is given the number 240, a mysterious number. The letters Mer, Mim, Rosh represent the number 240. Interestingly, as we peruse the American mathematical grid system of the pyramids, a great researcher, Karl Monk, uses this very number to decode pyramid connections, longitude, latitude, and grid math across the planet. The reason why this becomes important is because we are highlighting connections between the West and the East. Here, once again, from the pyramid text, the oldest writings, we see the apathy of the ancient Tamarians, misnamed Egyptians, for those in the south. The flame is extinguished. No light is found in the house of him who possesses Nubit. They were not from the south. The earliest texts say they were from the west. And the term that they used for the west, Amashi, was not native to their language. Interestingly, the She is the name for the oldest American civilizations. Here we have a map depicting hominids, non-Homo sapiens, by human evolutionists all throughout East Africa, with one point in Chad in the Sahara. As we continue to look at where these fossils were found, all of the data that we have provided before this time begins to put the puzzle together completely. East Africa shows that it was a place that suffered greatly from this cataclysm, and it shows up in the human biological polymorphisms that cause Homo sapiens to devolve, to the point where human evolutionists call them something that they are not. When we turn to Gondwana, we begin to see from the plate tectonics what took place. And this study provides with the beginner proof and evidence of what I need to substantiate the claim. Stratigraphy and paleobiology of the Upper Cretaceous Lower Paleocene sediments from the Trans-Saharan Seaway in Mali. What this paper discusses is an ocean in the middle dividing West Africa from its Central African regions. Yes, the Sahara is a desert today, but prior to it being a desert, the evidence shows it was a place where there was an ocean, animals, fossils. What we're seeing is this area was destroyed by some major event. We're discussing the event that caused this particular destruction and caused the loss of this life and the desertification of this area in Africa, which has the largest desert, the Sahara. Here is a depiction from the study that shows the types of animals that lived in this area. Now let us read from the data that the study gained. An epicontinental sea bisected West Africa periodically from the late Cretaceous to the early Eocene, in dramatic contrast to the current Sahara Desert that dominates the same region today. Known as the Trans-Saharan Seaway, this warm and shallow ocean was a manifestation of globally elevated sea level associated with the rapid breakup of the sub-supercontinent Gondwana in the late Mesozoic. Although it varied in size through time, the Trans-Saharan Seaway is estimated to have covered as much as 3,000 kilometers of the African continent and was approximately 50 meters deep. The edges of the sea were defined in part by the high topography of the Precambrian cratons and the mobile belts of West Africa. Over its approximately 50 million year episodic existence, through 6 major periods of transgression and regression, the Trans-Saharan Seaway left behind extensive nearshore marine sedimentary strata with abundant fossils. The waters that yielded these deposits supported and preserved the remains of numerous vertebrate, invertebrate, plant, and microbial species that are now extinct. These species document a regional picture of ancient tropical life that spanned 2 major Earth events, the Cretaceous Paleogene boundary and the Paleocene-Eocene thermal maximum. Whereas extensive Epiric sea flooded the interior portions of most continents during these intervals, the emerging multi-continental narrative has often overlooked the Trans-Saharan Seaway, in part because fundamental research, including the naming of geological formations and the primary description and analysis of fossil species, remain to be done. We provide such synthesis here based on 2 decades of fieldwork and analysis of sedimentary deposits in the Republic of Mali. Northern parts of the Republic of Mali today include some of the furthest inland reaches of the ancient sea. And here is a map of the depiction that they give in their rendition of what the sea would have looked like. What we are saying is they're just catching up. We are light years ahead of these geologists, archaeologists, and anthropologists who are putting this data together, because we have more information, more ancient information. Our maps tell the whole story, why the Egyptians said they were from the West, where we see on the map in D, why the hominids, the microcephaly hominids, the polymorphic degenerated Homo sapiens are found in section F. These questions now can be answered. We don't have to rely on ocean dispersals and weird theories of monkeys and dinosaurs swimming across oceans. The antiquity of humans is evidenced here. And now we will show where and why the biological polymorphisms began to devolve. Now let's return to the eastern part of Africa and give a thorough explanation for why they are finding all of these fossils of hominids, and they are believing that these hominids were precursors to Homo sapiens. They were not. They all were a part of geographic areas that experienced major cataclysms. So here you see in East Africa the dots depicting. Here we have a major catastrophe that took place. The Lake Toba explosion of a volcano. That volcano caused the areas that we are depicting to go through major environmental changes. This is the center of the blast, and this is archaeologists and geologists developing a map of where the ashes actually extended to. As we can see, the ashes reached all the way to Pinnacle Point, South Africa, caused a global decrease in the global temperature, and the areas impacted had sulfur aerosols that were released throughout these areas. Well, what did the sulfur aerosols do? They got into the plant life, they got into the air, which means they got into the organisms, especially the organisms who are in East Africa, Homo sapiens, who now are breathing in high levels of sulfur aerosols. What did that do to their biology? What did that do to the biology of the peoples in Asia, the peoples in India? The releasing of the sulfur aerosols depicted here by the scientists who developed the study shows that it was the second greatest catastrophe, leaving record in the strata of the Earth's crust and evidence of the volcanic ash. The sulfur aerosols passed through East Africa all the way throughout Europe. What do we find significant in this pathway? We find mutations in the MC1R gene. Yes, melanin, which is based on selenium and copper. But the pheomelanin phenotype is based on sulfur. So what we have here is an environmental, epigenetic, scientific explanation for how pale skin gained its sulfur epigenetic molecules. The alteration that it caused is the loss of pigment. The timeline we are dealing with is 50,000 years ago and 12,000 years ago for these great catastrophic events, which we will detail further. But the area is most important to pay attention to. Here is a study that shows that in Ethiopia, they were impacted by genes that were introgressed into the DNA of Ethiopians. This SLC24A5 gene, where there's a mutation, also causes skin to lighten. It explains why the sand bushmen have light skin, but also have DNA from the Africans who are in that area who are very old. So we have two gene pools that we are discussing here: the melanocortin receptors, MC1R, and the SLC24A5 gene. We must understand that genes work in groups to perform phenotype. So when we're looking at someone's melanin, we're looking at groups of genes working together to give that phenotype. Here, scientist Keith Ching identified the key gene that contributes to lighter skin color in Europeans and differs from West Africans. In 2005, Ching reported that one amino acid difference, which is a what? Frameshift mutation that we talked about earlier, in the gene SLC24A5 is a key contributor to the skin color difference between Europeans and West Africans. He's mentioning West Africans for a reason. All instances of a gene mutation that contribute to light skin color in Europeans came from the same chromosome of one person who most likely lived at least 10,000 years ago, according to Penn State College of Medicine researcher. The mutation in SLC24A5 changes just one building block in the protein and contributes about a third of the visual striking differences in skin tone between peoples of African and European ancestry, said Ching, distinguished professor of pathology. Now this is where we have to pay attention. This specific mutation in SLC24A5, called A11T, is found in virtually everyone of European ancestry. A11T is also found in populations in the Middle East, and Indian subcontinent, but not in high numbers in Africans. Researchers found that all individuals from the Middle East, North Africa, East Africa, and South India who carry the A11T mutation share a common fingerprint. Traces of the ancestral genetic code in the corresponding chromosomal region indicating that all existing instances of the mutation originate from the same person. Europeans have 10 to 20 times more instances of the melanoma than Africans. However, despite also having lighter skin, East Asians have the same melanoma rates as Africans, which means less. The reason for this difference can only be explained after the gene mutations for both groups are found. Removal of the NCKX5 melanotropic peptide disrupts melanogenesis in human and mouse melanocytes, causing a significant reduction in melanin pigment production. Site-directed mutagenesis corresponding to a non-synonymous SNP in SLC24A5 alters a residue in NCKX5. So what we're describing here is the frameshift mutation. The insertion of one amino acid shifts the whole frame, and now you can't produce the dark melanin anymore. And this is all contributed to environmental catastrophe, whether man-made or whether caused by nature, which is a whole discussion within itself. Regardless of the argument of whether it's nature or man-made, meaning nature, environmental catastrophes, or man-made, meaning catastrophes caused by man, the science stands. We are telling you where white people come from. We are telling you where others who have close genetic association come from for the first time. As we can see, the habitable areas least affected and most distant from the catastrophe and sulfur aerosols and toxins were North America, Central America, South America, and West Africa. These are the places that have the oldest Homo sapiens sapiens with the greatest genetic diversity and the highest levels of biological carbon in those Homo sapiens. Pale skin is a disease, and the ancients knew it. Now scientific fact show it's a mutation. It just did not know how the mutation came about. The ones who did tried to hide it under natural selection. They tried to make it an adaptation. All the extinct classes of hominids they labeled are proof of disease. Extinction comes about through disease and can be related to catastrophes in toxic environments that deplete resources, food, clean water, and inability to survive. It is a fact the paler groups are having a birth decline problem globally. And now we can take our conversation to haplogroups. This is a diagram showing the male Y haplogroups that we get from our fathers. These do not recombine with any of the genes, therefore they're stabilized and we can measure them from father to son to tell where someone comes from or who they're related to. This grouping of A, B, C, D, and E, F, H, G, ongoing, also gives us geography, worldwide geography of peoples and where they inhabited. Even after migration, we can see the admixture events and discuss the origins of people. But more importantly, the haplogroups themselves deal with the mutation rates that we were talking about. And this is the most important part that is not talked about. So when we're looking at the changes in the haplogroups, the changes are based on SNPs and STRs, SNPs being single nucleotide polymorphisms, STRs being short tandem repeats in the SNPs, so that we can compare the two. Here's a graph that shows the infertility rates as it's coordinated to the haplogroups. So what we're saying is that we can measure the antiquity of people based on these haplogroups. We can also measure the vitality, the fertility levels of the groups, as we are seeing here. When we move outside of the E haplogroups, the other haplogroups, which are primarily Eurasians, are weaker in fertility, experiencing azoospermia and other maladies that show up on the chromosomes. More importantly, we can help fix these mutations. That is the most important part of the conversation. We can tell globally the genealogy of all peoples now. We can tell what caused them to have these massive mutations in their genetics. And more importantly, from the Aboriginal Medical Association, we can help them fix these problems. But we have to solve the problem of who is the original man, who is the original woman. The biggest problem in modern genetics is using these two haplogroups, haplogroup C and haplogroups Q, to tell us who is native to America. This is a fallacy. The arrays that are used to measure for who is native to America are using arrays from Asian slaves from Asia who showed up in Acapulco, and from two other migration points that we have mentioned in part 1 of this documentary series. What needs to happen is the original haplogroups A, B, and E that were in America longer than any other haplogroup need to be the measuring standard for who is native to this land. Not only that, we need to highlight that there are certain small circles of people who know this information and who have worked through institutions to hide it. The reason why you are hearing this documentary is because the information was suppressed. We are only bringing out what is known by a group of people who don't want you to know it, who want to hide your identity. But they can't hide it. It's all around us. Take a look at this turtle or tortoise. What do you notice? The back has 13 groupings of shapes on the shell. And when we count those and we count the outer shells of many turtles, we get the numbers 13 and 28. We know that America is traditionally called Turtle Island because we see depictions of the she, or the Olmec Mayan Ahau, depicted with, in the art and in the cosmology, discussing the Ak, or the turtle. Why did they do this? Here we have the Paris Codex. The Paris Codex depicts what we call the Manzil calendar. The Codex shows the she had a 13-phase, 28 days per phase calendar. This is often called a Manzil calendar in other traditions, in India and amongst the Moors. Those other two civilizations also developed the same zero that the she had. The 13 by 28 gave us 364 days with an additional day to cap off the tropical year. But the Paris Codex matches a much older verification of this calendar. So we see it in nature, we see it recorded in the scripts of the Mesoamericans. Where else do we find this ancient calendar, and how is it associated with the discussion that we are having? We find this in South America, and as we peruse where we find it in South America, it begins to make everything connect. Here is the depiction of this calendar from the Ica stones. We see the warrior with a crescent on his head with 280 days, representing the calendar 28-day periods, a complete 10 of them. Then we see 84 squares near his face, representing the other 3 28-day periods, a total of 364 squares, representing again the Manzil calendar. What we are looking at here is a connection. The 8th continent being shown on the ecostones, the oldest calendar being shown on the ecostones. The American civilization is very old, but that antiquity has been suppressed, and it is directly connected to its oldest inhabitants, who are now called Black Americans, Afro-Brazilians, and other groups of people who are here. How long have we been here? Well, let's just ask our ancestors. Each of the years called a tune by the Maya or the Shi in these dates is composed of 360 days. If we return to the creation date, which is 20 cycles set at 13, we see that it will take 41 octillion, 943 septillion years from the moment that they say that their ancestors manifested from the back of a turtle. The mathematics, the calendars, and the history here is remarkable. What's hidden is that these Shi were dark-skinned people with locks who depicted themselves, illustrated themselves in their own temples, being invaded by Asians and in being invaded by Europeans. Here are the 13 ancestors depicted in a divination system that are connected directly to the calendar. No, we don't have to make anything up. This is from the Temple of the Warriors in the last period of our rule in Mexico before we regained, and the Asians and Europeans were on the walls of the Temple of the Warriors at Chichen Itza. But the antiquity in the Americas has been ignored or suppressed. Here, a New York Times article states, "Find skull traced to earliest man. Dr. Wolf discovers Patagonian fossil said to be millions of years old." Buenos Aires. The discovery of a fossilized human skull of the Tertiary period was announced today by Dr. J.G. Wolf, who has just returned after 2 years of exploration in Patagonia on behalf of the La Plata Museum. The article is dated February 27, 1923. The author states, Buenos Aires, February 26, Associated Press. The discovery of a fossilized human skull of the Tertiary period was announced today by Dr. J.G. Wolf, who has just returned after 2 years of exploration in Patagonia on behalf of the La Plata Museum. Dr. Wolf said that the fossilization was that of the Tertiary sandstone and that there was no doubt it was formerly the skull of a human being, not an accidental formation. The lower jaw is missing, but otherwise the skull is almost perfect. Let us keep in mind that the Tertiary period is between 2 million to 66 million years ago, and they were measuring by stratigraphic evidence. The article goes on to state, the fossilized human skull, believed to be of the Tertiary period, which was unearthed in Patagonia and reported at Buenos Aires by Dr. J.C. Wolf, will, if it proves authentic, antedates by hundreds of thousands of years any human relic thus far discovered. Dr. Frank Boas, anthropologist at Columbia University and world-famous scientist, declared to the Associated Press last night. Keep in mind, Frank Boas is a teacher of who? W.E.B. Du Bois. The article continues. It will be even older, possibly 500,000 years, said Dr. Boas, than the skull of an erect man-ape known as Pithecanthropus erectus, itself 500,000 years old, which was found in Java 29 years ago. The anthropologist expressed amazement at the news of Dr. Wolf's reported discovery. He had received a letter from his friend, the explorer, only yesterday, and it contained no mention of such a valuable find. If it is definitely established that man existed in the tertiary period, it will upset all accepted scientific views regarding the American continents and the antiquity of man, said Dr. Boaz. The discovery, which seems almost too marvelous to be true, will throw light upon this hitherto unknown period, Dr. Boaz said. So far, scientists have merely speculated upon the possible existence of man in the tertiary period, although traces pointing to the existence of a man-like animal have been found in various rocks. Here is a depiction of the Brontosaurus skull as it appeared on page 309 of the Congressional Serial Set of the United States Governing Printing Office. But we had a problem. We had Charles Darwin developing his theory of natural selection, and the world went with natural selection. What we have proven today is natural selection is wrong. And now we have to revisit the antiquity of the Americas. Here is one scientist who tried, Florentino Ameghino. He is known for the development of cladograms and phylogenetics. Studying history shows that his discovery was stolen from him, and there are many papers written on this. Ameghino talked about the antiquity of the Americas, gathering many fossils of humans, and developing a system of evolution as he saw it in the human family originating in the Americas. While I do not agree with everything Ameghino stated, I do agree that he was on the right track. Other scientists tried to stand up for this reality as well. Dr. McIntyre dated a site at Haplogroups to 250,000 years ago, with evidence of humans using tools and leaving their marks on camel bones. Uranium series dating was done, and zircon fission dating was done at the Haplogroups ash deposits, 270,000 to 245,000 years. But this is a problem for modern human evolutionists because it doesn't fit into the out of Africa or the out of Asia hypothesis. Therefore, the rule is dismiss it. But we know for a fact that you cannot dismiss it. The evidence is all around us. The antiquity of our use of higher mathematics is proven. Primordial science gives us a good idea of how this works. In our next episode, we will talk about the pyramid system. Here we have Cholula and the Great Pyramid systems. These systems were in use of a very ancient technology that developed longitude and latitude and gave us global chronology. Here is our thesis on global chronology. Here are my 9 successive magnetic pole shifts. My timing does not match or agree with conventional dating. I will have to cover that in another discussion. The 9 successive pole shifts are written in nature, in our environment. There are 2 that we want to give attention to here. The importance is that the prime meridian had to be at Cholula or Tiwanaku in South America and then spread to Giza in Egypt. This reality is covered in when and where we built pyramids in a global network. Here we see the 9 magnetic pole shifts that are recorded on the seafloors of the oceans. Geologists have confirmed these 9 magnetic pole shifts, although their dating covers it over a 4 million year period. The dating here is unimportant. What's important is that it falls within the Cataclysm thesis. And we have environmental proof of it all. Here, 50,000 years ago, Europe was on the equator. We have the evidence to show that Europe, underneath its layers of soil, has proof that it was once a tropical area. At this time, the Laurentine ice sheets that we showed earlier in parts 1 and 2 covered parts of North America. The northern spin pole at the time was 17 degrees from its position at the Okpatok Island in the Greenland area. This alignment matched perfectly with the Cholula Pyramid, proving that pyramid had to be older than 50,000 years old. It also proves that the civilization around the Giza Plateau moved into that area around 50,000 years ago. These alignments from Tiahuanaco in South America and the Xi'an province in China also show an alignment to the Okpatok pole, meaning that they also are over 50,000 years old. We can use this grid pyramid network to describe the origins and the cradles of civilization across the earth. The pyramids in Tiahuanaco are on a perfect 180-degree alignment to the pyramids in the Xi'an province in China. That alignment can only be seen if the Okpatok pole of 12,000 to 50,000 years ago is used in the measurement. These poles are verified via the study of hysteresis of the volcanic rocks, as we have shown from the maps of the 9 magnetic pole shifts prior. The argument of when is an issue of geologists versus credentialed geologists and the scientists that are out of the box. The issue is, who built these? The answer is given and is documented, and the monuments are left for us in stone. Here we have the Xi'an province pyramids and the sunken pyramids we showed earlier from the 8th continent, being a distance of 16.34 degrees from each other. This is important because Tiahuanaco is exactly 16.34 degrees from the Ice Age equator, or the equator that existed from 12,000 to 50,000 years ago. This system of mathematics will give us all of the data that we need to show us the cradles of civilization. These particular spots, which match the pyramids that we are discussing, all point us to an ability to track global genealogy through genetics and through the archaeology left and the building of civilizations. Through this, we will be able to measure where peoples come from, using the haplogroups and using these cradles of civilization. Specifically, we will be able to highlight who is aboriginal to the Americas and who is not. Not only that, we can trace these peoples back to the original locations that they developed in. This entire series was about giving people the scientific ability to track where they are from, who they are from, and even improve upon the things that have happened based on environmental and human-made catastrophes that have contributed to the complexity amongst the human population. Our next part will deal with this. We start our conversation off with a discussion on the origin of Native Americans. Earlier in the series, I talked about 3 waves of Asians who came to the Americas: the Toltecs, the Aztecs, and the Manila Galleon, which brought Asian slaves to Acapulco. Here, I will delve deeper into the genetics that support this statement. There were no Native Americans that came across a Bering Strait. 26,000 years ago, 13, at no time. The source population came on boats. 2 voluntarily, 1 by the slave trade, the Trans-Pacific slave trade. Here, I am showing for our memory the Temple of the Warriors at Chichen Itza, which depicts these Asians, who were the first from Asia to come to the Americas. And who did they find? The primary population of black people, indigenous people, who are the autochthons of this land. Here, we have a study published at the National Library of Medicine. Mitogenome evidence shows 2 radiation events and dispersals of matrilineal ancestry from northern coastal China to the Americas and Japan. Let us read from the article. Although it is widely recognized that the ancestors of Native Americans, primarily came from Siberia, the link between mitochondrial DNA lineage D4H3A, typical of Native Americans, and D4H3B, found so far only in East China and Thailand, raises the possibility that the ancestral sources of early Native Americans were more variegated than hypothesized. Here we analyzed 216 contemporary, including 106 newly sequenced D4H mitogenomes and 39 previously reported ancient D4H data. The results reveal two radiation events of D4H in northern coastal China, one during the last glacial maximum and the other within the last deglaciation, which facilitated the dispersals of the D4H sub-branches to different areas, including the Americas and the Japanese archipelago. The coastal distributions of the Native Americans D4H3A and the Japanese lineages D4H1A and D4H2, in combination with the Paleolithic archaeological similarities among northern China, the Americas, and Japan, lend support to the coastal dispersal scenario of early Native Americans. Here is a map of the dispersal of the D4H lineages, one to an area called Hokkaido in Japan. These are the lineages that eventually show up in the Americas. But there's a bit of a question that has to be asked and must be demanded for anyone viewing this. These Homo sapiens would have had to have boats. How else would they have dispersed to Japan? This is a visual of them. Now, we have to ask the question, and we beg the question, if they had boats to travel to Japan to disperse these mitochondrial lineages, did they have boats to disperse these lineages to the Americas? We are led to believe that ice had to freeze in order to create a pathway for those in Asia to get to the Americas. Here we have living proof of genetics spreading across water. Therefore, we know they did not swim all of that area, so we conclude that they had boats. This is exactly what is depicted in Codex Barterini. Groups of people coming on boats. Also in Codex Ascatitlan, we see the very same thing, depictions of Asians coming on boats to Mexico. We follow that up with the Manila Galleon, which traveled from the Philippines and the areas of the Southeast Asian islands to bring slaves over into Acapulco on boats. This D4H3 genome that passes from China and Japan into the Americas has no way to get here but via boats. As we stated, you cannot get from mainland China to Japan without these boats. So we know for a fact that these people, regardless of the time period that we are talking, used boats. The evidence is in the codexes, is in the history of slavery into North America of Asians. Now let's divert this conversation to discuss bottlenecks. Here is an article, Genomic Inference of a Severe Human Bottleneck During the Early to Middle Pleistocene Transition. This becomes important to our conversation as we have discussed the major cataclysms that hit the planet. Each time it caused bottlenecks. A bottleneck is when the population size shrinks down to a very small size due to the cataclysm. Then we can find out from the bottleneck who survives the bottleneck, because those people will be still present on the earth as we have it today. Population size history is essential for studying human evolution. However, ancient population size history during the Pleistocene is notoriously difficult to unravel. In this study, we develop a fast infinitesimal time coalescent process, FITCO, to circumvent this difficulty and calculated the complete likelihood for present-day human genomic sequences of 3,154 individuals. Results showed that human ancestors went through a severe population bottleneck with about 1,280 breeding individuals between around 930,000 and 813,000 years ago. The bottleneck lasted for about 117,000 years and brought human ancestors close to extinction. This bottleneck is congruent with a substantial chronological gap in the available African and Eurasian fossil records. Our results provide new insights into our ancestry and suggest a coincident speciation event. Notice that they did not use fossils to come to this evidence. They used a study of living people, 3,154 people. The lack of fossils is what gave them the indication that something was wrong. The population dropped down to 1,280. But if you look at this graph, you will see that the predominant makeup of the population is African, with non-Africans branching off later. Here, African is based on the genome and not fossils. Therefore, we know from the study that Africans and African Americans were used. The question we pose is that during this bottleneck and post this bottleneck, the survivors, what do we know about their genetics? And why did we test modern genetics to give us an indication? Well, we do know that the oldest haplogroups would have survived the bottleneck. And the question is given, what is the oldest male haplogroup? It is haplogroup A. Haplogroup A is found in the Americas and in Africa. As we have stated earlier, none of our alleged hominids have haplogroups at all. Not one. Because there's never been DNA collected from them as fossils. The only evidence we have of antiquity is in the genetics of modern black people. But the survival of this particular cataclysm and bottleneck begs another question. Who has the oldest DNA on Earth today? And when we answer that question, we answer a lot. That DNA for the Y chromosome is not found in Africa. It is found in America. Here is the DNA of the Perry family. Albert Perry's family, who tested 3 members, do not have the same DNA as Africans in Cameroon. They have S&P variants that are not present in Africans in Cameroon. S&P stands for single nucleotide polymorphisms. Here is a chart that shows the exact variants and the differences. The DNA here in America is older. So as we look at this and we know that we have a different variant of the oldest haplogroup amongst black people in the American Southeast, we beg ourselves to ask a question again. How did it get there? Did it come through slavery? We use the records to find out. And we find out that Perry was born in Florida in 1816. But in 1816, Florida was Indian territory and Spanish territory. At the time, Perry would have been born amongst the tribe or confederation known as the Yamasee that became the Seminole Confederation. This lineage of blacks, which goes back to the pre-Muskogean aboriginals of the Southeast, the Ahashita or the Hichiti, have direct lineage to the Shi and the Mayan in central and lower parts of North America. And as we have stated, this is the lineage of the oldest lock-wearing, dark-skinned, pyramid-building, mound-building peoples in North America, who had bases in South America and in North America. Here is the lineage of Perry. He was never listed on any censuses as a slave. So why are they saying that he was a slave? It's a comfortable way to explain away the truth. The truth is that what we are discovering in this time is that the lineages here are older. Here's a map of these lineages. Perry would be in the A stock to the left, which later branches off into the other mutant groups. And I stress mutant, not to insult, but to say that the Y chromosome of these individuals went through drastic frameshift and missense mutations that we can chart today. They are not old. The bottlenecks and the people who survived the bottlenecks would be classified in the A groups. So what are you saying, Ali? I am saying that blacks in the Americas are the oldest people on Earth. The genes prove it, the heritage proves it, and the history proves it. And that we can chart the mutations off from this group for all populations. Remember, these mutations are not natural selection or trait selection. These mutations are the point mutations that we mentioned earlier, the missense mutations, and the frameshift mutations. Viruses, bacteria, pathogens, and environmental catastrophes led to the shifts in the population, the A at the bottom being the primary and the R being the European. So when we ask the question of who is the original man, we get an answer, a very definitive answer. Europeans want to make themselves the original men. They are using evolutionary biology in the sense of human evolution to tell a story that never happened. And they're coupling this story with layers and layers of fallacies, where Asians become the first Native Americans. Pale skin, hairy beings become the first beings on Earth. And the curriculums are never questioned. This documentary series is about questioning those things. You are looking at how the Aboriginal Americans, the Indigenous Americans, the autochthons in America depicted themselves in art. Let us ask a question. They told us that Columbus discovered America, but they never showed us these Americans. Why do you think that is? This story that we are telling is laying a bedrock for a new conversation about global genealogy and global genetics. It will be the precedent conversation that leads all other conversations in the future about global genetics and global genealogy.